In this video, we're going to introduce you to polymerase chain reaction. Now, polymerase chain reaction was a revolution in the biotechnology field, and it addressed a very, very important problem, and that was access to enough amount of DNA to do analysis. And very often, there were times, especially when there was a need to solve a crime or analyze samples in which very little is available, that created a problem because there wasn't just enough DNA to do any analysis. So polymerase chain reaction made it possible for scientists to, even theoretically, with starting with one molecule of DNA, they can create billions of copies of that same DNA and do analysis. So polymerase chain reaction is just a series of steps that are repeated over and over and over again. And it relies on the ability for hydrogen bonding between pieces of DNA. And it also is on the basis of DNA replication that we learned earlier. So what do you need for a polymerase chain reaction? I'm going to write it here. First of all, you need your DNA template. Okay, let's say you don't have very much of this DNA template and you want to amplify it. So that's your DNA that you want to amplify. And then this DNA is going to be, so the DNA is hydrogen bonding. There's lots of hydrogen bonding between bases, just like shown in here. Okay, so then what you need, you're going to follow the steps of DNA replication. And if you remember, for DNA replication, you need those little primers. So in polymerase chain reaction, you also need these little primers. So you need primers. And since we know the sequence of the DNA we want to amplify, we can design the primers. And then we need an enzyme, a DNA polymerase, that does the job of the polymerization. DNA polymerase. And then we need the starting material, the basic material that you need, and that's nucleotides. So I'm going to write as DNTPs, deoxynucleotide triphosphates, which you can use to build nucleotides together. Okay? So, and there are 3 steps in polymerase chain reaction. And first is denaturation. So you treat the DNA, your sample, at a very high temperature. That's called the denaturation steps. And you separate these strands. The next step is annealing. And that's where the primers that you designed are going to hydrogen bond with the DNA template. And for that, you need to reduce the temperature. And really the temperature depends on how big these primers are. And these primers are usually maximum 30 bases. So then once the primers are annealed, now you have these 3' hydroxyl ends. Remember we said for DNA polymerase, for any nucleotide to extend, you need an available 3' hydroxyl. So then these 3' hydroxyls are used during the extension phase of PCR, where this piece of DNA is extended. And then now, after just one round of this denaturation, annealing, and extension, you started with 1 double helix, you ended up with 2. Now this process is repeated about 30 times. Okay? And if you can think about it, you double the DNA material and you do it 30 times. And this is a huge number. So after going through the steps, denaturation, annealing, extension, denaturation, annealing, extension, you're going to get millions of copies of the DNA that you started with. And that basically, it's a very simple process. And when it was developed, it basically allowed us to do lots of different things. So here's an assignment for you. Here's a double-stranded DNA sequence. Design primers to PCR amplify the DNA, and then show the steps of the PCR cycle. What would they look like? Denaturation, annealing, and extension.